Okay, so hello everyone. Welcome to this another of our trainings and talks for Project Accessible Genomics. Our speaker today is Joe Russell. Joe is the chief scientist at the MRI Global, and he is also a kababayan by marriage. His wife is of Filipino descent. He has been here in the Philippines, although he has not been to Mindanao yet. So I hope that after this pandemic, he could visit us and we could do like training in the beach or something. All right. So please welcome Joe Russell.
All right. Yeah, thank you for that introduction. And yes, would love to take you up on that. Um, it's been quite some time. It was 2007 that I was last there, but I would love to get back. So thank you all for tuning in, and appreciate you giving me the chance to speak with you today. When Khalil reached out, I mean, we had discussed a few things to talk about, and just one of the things that's been on my mind lately through my work at MRI has been wastewater. So doing genomic surveillance for SARS-CoV-2 in wastewater. We haven't begun this process operationally yet, but we have written several proposals and we're waiting to hear back. Hopefully we will actually be actively doing this in the Washington, DC area soon, in the next few weeks, actually. So we've just been thinking about it and thinking about standing up, surveillance operations, both from just your standard PCR, but also from a genomic standpoint. And so I wanted to go through that and discuss it sort of in the context of a lot of my previous experience with nanopore sequencing, which has been through developing laboratory platforms to enable use of it outside of a traditional laboratory environment, so out in the field. And so, yeah, I'll just go ahead and start. So a little about me before we dive into it. As Khalil mentioned, my wife is half Filipino, and we did make a trip there in 2007. This background photo that I have on my slides is actually I took on a hike outside of Sabang in Palawan. So we were there in 2007, visited her family in Lucena, just outside of Manila, and then family in Cebu, and then we went all around Bohol, Siargao, was the closest I got to Mindanao, and then Palawan, and a little bit around northern Luzon. So yeah, definitely excited to get back at some point. As far as my scientific background, so I have a PhD in geomicrobiology. So that is essentially the study of microbial life and how it interacts with the lithosphere, so the Earth, rock crust, geological environments. I did a PhD based on studying microbialites, which are these sort of carbonate accretions that are built by microbes. Actually, some of the oldest fossils of life on Earth, 4 billion year old fossils, are actually these types of structures that were found in Australia. And there's only a few places on Earth where they're still growing, and one of them is this lake in British Columbia, Canada, that we got to go study. And then also looked at samples from the subsea floor. So most of my PhD was in subsea floor microbial ecology. So it involved a lot of field work. I used molecular and cultivation-based methods to look at these environments. So that was, cultivating microbes and also doing PCR and metagenomics. But my project was collecting these samples in the field and then bringing them back to a lab. And a lot of what I did in my postdoc at MRI was trying to go out into the field and apply those same methods I was using in grad school, but do them in the field and in a very different context. So I did a postdoc at the MRI Global, which used to be MRI Midwest Research Institute. A few years ago, they rebranded to MRI Global. I'm in their just outside of Washington, DC office, their biosurveillance office, in the Global Health Surveillance and Diagnostics Division. So we do a lot of biosafety and biosecurity training, lab management and operations, and infectious disease and biological threat detection, R&D, research and development. And one of the big programs we have ongoing is our mobile laboratories. And so during the West Africa Ebola outbreak, MRI Global had a footprint in 3 countries, so Guinea, Sierra Leone, and Liberia, where we had our mobile laboratories where, they were mobile in the sense that you could put them in the back of a C-17 and fly them over to the in country and stand them up and have them operational in 72 hours. And this was this was one of our lab in Moyamba in Sierra Leone. And you can see sort of the footprint of this lab as it expanded once it was there in country. You had your master mix PCR module, decon and shower, sample receipt module. So it sort of expanded into this sort of mini campus. But that was our experience with mobile laboratories through the Ebola outbreak. I joined MRI in 2015, which was when the outbreak was sort of winding down, but I did get a chance to go to Sierra Leone after the outbreak was over to continue our molecular diagnostics training. And so this is myself in 2017 in Sierra Leone with our trainees. But one of the things that we noted when we were over there was, and speaking with people that were there in country during the crisis, was, we would have a mobile laboratory in place, and we would hear of a patient that was 200 miles away, and it would be rainy season in Sierra Leone, and the roads would look like this in rainy season. So the likelihood of that sample from that patient getting to that mobile lab was, very low. And so this was around the same time that these, at the time they were emerging. They're sort of matured a lot now, but these, ultra portable handheld molecular biology tools were sort of coming online, allowing the capability of these advanced environmental biosurveillance workflows and clinical diagnostics workflows far away from, your traditional brick and mortar lab. So on the left, you have a Biomeme qPCR thermocycler. So this is a actual real-time thermocycling device that runs off an iPhone. Biomeme is a startup company outside of Philadelphia in the States. And then, of course, everybody on this group is familiar with the Oxford Nanopore's MinION. And so this really sort of changed our thinking about what a mobile laboratory could be. I'll get into this a little bit later after talking about wastewater surveillance, but one of the things when you're trying to do this out in the field is that although the device footprint is very small, the operational footprint of doing this work in the field is not equally small. And so just because your sequencer is the size of a candy bar, you end up going out in the field, but you need cold chain, you need a generator, you need a place to work, you need all of these things end up ballooning your footprint into this sort of, it takes away from the full realization of the MinION and its real-time capability and its small footprint. And so, this is kind of the ideal that we're trying to strive for, where you're just, you can load a flow cell on the beach, go for a swim, come back, your samples are sequenced, you just, look through your data while you're getting a tan. But often, if you pan the camera to the right or left in that first photo, you'll probably see something like this. There's a lot that goes into it. And so that's sort of the context of how I've been thinking about using MinION is getting it out in the field in a sort of outside of the traditional laboratory environment. And I'll come back to discussing that in the context of SARS-CoV-2 and specifically wastewater surveillance. But right now, what we're looking at is just doing that for, in our brick and mortar labs that we have here in the DC area. Right now, throughout the outbreak, it's primarily been accomplished through, RT-qPCR methods. But the utility of sequencing for variant detection is being explored by several groups now. And there's been in the past few weeks, there's been a few preprints and publications that have come out showing that it is indeed possible to do variant detection of SARS-CoV-2 from these wastewater samples. And so this was a preprint where they looked at samples in Switzerland and from, I think it was 3 different sites. And they were able to get really robust detection of the 2 primary variants of concern that were circulating in that area. And then I saw this one just this morning, I think it came out yesterday, where they actually did shotgun sequencing. So it was the first one in Switzerland, they were doing the Arctic protocol. So I think most of you are familiar with that from the videos I've seen on the training section of the Accessible Genomics website. That that is an amplicon based, where you're amplifying all these different small regions of the genome, and you can stitch them together after the fact. This preprint actually just did a shotgun sequencing, where they didn't do that amplicon sequencing protocol. They just went straight for sequencing directly the RNA or the cDNA, but still were able to get variant detection. And so, of course, I'm sure everybody's familiar with this by now, why this is important. These variants are, they really matter. If there's these changes in these particular sites on the genome, that can have a big effect on the efficacy of SARS-CoV-2 binding to its target cells in the human body. So that has an effect on it also can have an effect on transmissibility. And so this was I put the link there. This is a great series of talks, if anybody has interest, on some of the latest data coming out of South Africa, just talking about putting in context some of these mutations, specifically talking about the 501Y and the 484 mutation, and talking about how specifically the 484 in the receptor binding domain has been able to change the polarity. So that just slight tweak it meant from a negative residue to a positive residue, and positively charged residue. And so with the negatively charged human cell, it binds much tighter and is able to basically stick to its human target cells much more effectively. And so we're seeing this variant start to take over, and concerningly, it's also been shown to evade this convalescent serum samples from folks who've been previously infected. So the concern is that it can have this, essentially immune escape, and limit the efficacy of the vaccine, or we might have to do a quick redesign, and, on the current vaccines. And so it's really important to understand what variants are circulating, what their prevalence is, and if you can catch it early and isolate it to a specific area, and engage, inherently limited public health resources that much earlier, it just really would have a substantial impact on public health. And so just we've been thinking about implementing this in the DC area, and I've been spending a lot of time thinking of the various sort of steps in this process, the things that you need to consider before you embark on this, so you don't get tripped up on something you didn't think about. This isn't comprehensive. As I mentioned, we have not yet implemented this, so I'm not speaking from operational experience. It's just been a lot of my reading and planning for our hopefully imminent experience. But there's these couple things I've identified, and other folks have identified in the literature, that really need to be considered. So supply chain is a big one, just from molecular diagnostics in general, but certainly from a sequencing perspective, getting reagents, having that line of reagent shipments in place and unbroken is a big challenge, especially when now worldwide the supply chain logistics has just been heavily taxed under the current pandemic situation. Your sampling method, so how exactly are you sampling the wastewater? The sample storage and transport, viral concentration, RNA extraction, how you're getting the genomic material out, what kind of assay you're running and what platform you're running it on, and then reporting of results. So it's not just a simple sort of yes no detection. You want to be able to compare that detection across the board with other groups around the world, have your data be comparable. And so there's a lot that sort of goes into that up front before you start doing this, in terms of getting your standard curves and understanding, when you're out in the field getting a sample, how does that compare with what you expect and other samples that have been seen globally. So for supply chain, what is the availability of key reagents, hardware, consumables? this is going to fluctuate for each element of the workflow, but there are redundant options at key steps. There's more redundancy at certain steps than at others. But if you can plan some of that redundancy before you get started, that's a way to just sort of get around these supply chain issues. It's not, you know, it's oftentimes we optimize a protocol and we want to stick with that because that's what we've validated. But certainly in the current environment, it's good to just have, you know, backup options and be comfortable, you know, switching in other options when you can. So the sampling method, the two big ones that you know, for wastewater surveillance are grab sampling versus composite sampling. So grab samples are essentially what they sound like. They're just taken at a single instance in time, whereas composite samples, they're a series of individual samples over a time period, usually 24 hours. It's been shown that composite samples yield a more consistent signal with much lower variability between the replicates, but grab samples, for whatever reason, have often shown a higher viral load than composite samples. So there's a bit of a trade-off there. It just sort of depends on, you know, your access to the site and your ability to sample. And then, of course, timing of samples. So for wastewater surveillance, you know, it's a inherently human-linked set of samples. And thinking about sort of human behavior before you go sampling and thinking about your timing is something to consider. The papers that I've seen, and I can email out the references that I've been reading to get, you know, put this together. But they talk about, at least in the States, about how the heaviest wastewater production is in the morning and evening hours. So, you know, folks getting ready, showering, going to the bathroom in the morning, and then often, you know, there's an uptick in flow at night. Folks going to bed, washing up, going to the bathroom before bed, et cetera. So the timing of your sampling can impact, you know, your likelihood of getting good coverage for virus. So sample storage and transport, I mean, I think everybody knows it's good to keep things cold, especially RNA, which is you know, inherently more unstable than DNA. 4 degrees is effective if that's your only option for short periods of time, but it's just sort of generally recommended to, you know, conduct your analysis within 48 hours if 4 degrees is your only storage option. Of course, if you have a -80 or even a -20, you know, that's better than nothing. But it was interesting to note that -20 should be, several references I saw said it should be avoided because of the development of crystalline ice structures at that temperature can actually degrade RNA more than I guess at -80. So concentration. So was that, I'm sorry, was that a question? I couldn't hear who or see who that was, or was that just somebody unmuted?
I think it was an accident, but if it was a question, just a message.
Yeah, throw it in the chat. So next element, so concentration. So you grab a sample, you're going to want to try and concentrate your viral signal as much as possible before you move into molecular methods. You know, you don't want to be wasting a lot of flow cell space on you know, stuff that you don't care to sequence. And so the more you can concentrate that signal, the bigger bang for your buck, essentially, that you're going to get out of your either PCR reagents or sequencing. And so there's several methods to do this. Polyethylene glycol precipitation, ferric chloride precipitation, there's all sorts of stuff, skim milk flocculation, which I actually had some experience with in grad school for geological samples, glass wool filtration, ultrafiltration, ultracentrifugation. One of the things is there's not a really great sort of apples to apples benchmarking study for these types of methods. There's a lot of papers that have, you know, used various different methods and seemingly effectively But I think that's sort of a gap right now, is to understand which options are best for which types of viruses and types of pathogens, really, is it if you're working with bacteria versus RNA viruses, DNA viruses. But generally, there are some good solid methods that seem to work pretty well specifically for viruses. So using an enveloped coronavirus, so murine hepatitis virus, the absorption extraction method was shown, paired with centrifugal filter devices, was shown to have the highest performance yield in a reference that I'm happy to link. And PEG concentration was shown to have increased effectiveness when processed. You process both the liquid and solid fraction together. So that's something to think about. That's a little bit more of a laborious protocol, but it could be a good backup, like for for that redundancy for supply chain issues. So viral RNA extraction. So three main techniques: the organic extraction with solutions such as phenylguanidine isothiocyanate. The more common sort of kit-based extractions are the silica membrane spin columns. I'm sure folks are pretty familiar with those. They seem to be they were earlier, a few months ago, they were tougher to get. I think we've stocked up and they're a little bit easier. They're on the shelves now and easier to order, but that was definitely a bottleneck for a lot of folks. That were sort of, there was pipette tips and extraction kits were worth their weight in gold. Paramagnetic particles is another technique. Organic extractions can be done with more raw reagents, and so again, from that functional redundancy, that might be a good option if you do find yourself going to a website and all the silica spin column kits are sold out. It can be done, and so it could be an option, but again, a little bit more time intensive and laborious. Okay, and so RT-qPCR methods. So there's a lot of assays out there. The CDC, US CDC's assay, the WHO assay, targeting various regions of the genome. So N2, N3, nucleocapsid, ORF1ab, RDRP, spike protein. Of course, for some of the variants, as of late, we've been using these assays. The spike dropout as an indication that you have B117 variant. So depending on what assay you're using, it could be actually used to detect different variants. There's limited quality control data for SARS-CoV-2 assays run on wastewater sample matrices. A lot of these methods have been published quickly. There's still preprints. So there's not a lot of heck of a lot of validation. So really it's up to you. But probably prudent practice would be to run the SARS-CoV-2, if possible, run the assay that on the platform it was developed on or optimized on, just to limit those possible things that could go wrong. And then reporting of results. So this is really one of those things where the qualitative versus quantitative nature of these results. There's just your basic CT values versus a more robust sort of quantitative gene copies per volume of sample, which is really what folks have been sort of striving for. So we have a common standard, because CT, there's a lot of inter-run variation to CT values. And standard curves are difficult to determine from highly variable sample matrices like wastewater, where the distribution of the virus is not homogeneous, and you could have a lot of particle association biases or other confounding variables. But wastewater is one of the most challenging and dirty samples, and so specifically for this, it would be good to do that upfront work where you get really robust standard curves using spike samples across a concentration gradient, from multiple days, sampling points, volumes, before you start your routine monitoring, so you can achieve that. What linear dynamic range looks like, R squared, your expected precision, sensitivity, and LOD. That should be established up front. So then you can get the standard reporting of viral genome equivalents per volume of sample, which would be more comparable, across globally. Just the global community, we can talk. We can compare notes easier, essentially. So this is that that Switzerland paper. I pulled out one of the methods, some of the method sections, and you can see here they touch on each one of these points in this paper. So they talk about, you know, composite versus grab samples, their sampling method. They talk about sample storage. They talk about filtration. They talk about you know, the concentrating and their RNA extraction. So you can see they use the Kitajima M viral RNA mini kit. That's, you know, one of the sort of standard off-the-shelf silica column-based extraction kits. And then you can see, you know, the methods they went for sequencing. So they did cDNA transcripts were amplified using the Arctic protocol. And so, you know, that's, again, those series of amplicons that sort of essentially tile across the genome. And then they were sequenced on the NovaSeq rather than the MinION. And so they were able to sequence really deeply and with 250 base pairs, but millions of reads each, as opposed to the lower depth you typically get with nanopore. And you can see, this is figure 1 from their paper, looking at the occurrence of the B117 variant versus the 501. And you can just sort of see how patchwork, you know, even if you have as stringent of sampling as they did, you know, it's still difficult to do this variant detection. So, you know, the blue color shading encodes the estimated frequency of mutations detected in the sample. Pink indicates an absence of the mutation, so the standard strain, and white indicates missing values. And so these are the various, so A is the mutation, all the mutations associated with B117, and B is all the mutations associated with 501. And you can see the patchwork sort of distribution there of detecting the variant. And so just, it's important to keep in mind that each one of these times you take one of these samples, it is just a snapshot in time. And so, you know, it's very sort of, you know, the more resolution you can give to this sort of pixelated image here, the more sampling you can do, obviously the better picture you'll get. But certainly something is better than nothing. So they were able to get robust detection and identify these variants that were circulating in this area in Switzerland. And then one thing to think about if you're doing Arctic too is that you don't have to look at every amplicon. So if you know there's been strains identified that, and you know where those variations, those mutations are, where those loci are, you can just look at the individual amplicons from the Arctic panel where you know should be amplifying that region. And so this is one of the tables from their preprint where they just broke it out by those specific amplicons that had those key mutations associated with those variants. And so in terms of optimizing the costs associated with doing this, you don't have to waste flow cell space on amplicons sequencing amplicons that aren't necessarily informative. If your goal is variant detection, you can be a little bit more frugal with flow cell space by limiting it to the amplicons in the panel that cover these informative regions. And so I guess coming back to what I was discussing in the beginning, we've been thinking about this, how to start doing this in the DC area. And when we do that, we'll likely be doing it in our brick and mortar laboratory just outside of DC. But my particular interest is how do we enable this in resource limited settings? How do we enable this in the global South? How do you utilize the real-time and portable nature of MinION sequencing, so you don't have to wait to send samples back to the laboratory? You can get this data, you can get this detection of variants in real time. How do you do this at the point of sampling? And so what, I've spent a lot of time thinking about and developing things to assist in this, process of getting these technologies further afield, is our platform that we developed called Mercury Lab, that takes advantage. We designed it around these emerging, now that much more mature from when we started, portable handheld molecular biology tools. So the BioMeme and the MinION. How do we support those things in the field? Again, with that ideal versus the reality, it would be nice to land somewhere in between, these two. I don't think we're going to get to that ideal anytime soon, but somewhere in the middle, I think, can look something like this. So this is our Mercury Lab platform. This is what we've arrived at now after 5 or 6 progressively less ugly prototypes. And that we started this about 5 years, 4 or 5 years ago now. But we've arrived at this point, basically a platform that allows us to take the laboratory to the sample rather than the other way around. And but having all of those things that you need to do these genomic workflows built in and supported. And so one of the things, some of the things you need to think about if you're going to try and do this in the field is power. Is really, if you're going to be sequencing, that drains a laptop battery very quickly. You're going to need more than just a laptop battery. So one of the things, originally we were going to pair this with a lithium ion battery brick, but those don't travel well. Once cell phones started exploding on airplanes, no airplanes want to take it anywhere. So we ended up just going with a simple AC/DC power inverter, because the power format that is cheaply and widely available across the world is 12-volt car batteries or 6-volt motorcycle batteries. And so through the inverter, you can directly power the lab through those easily available power sources. And also the inverter it can directly run off of solar energy with a third-party solar energy capture device. You can plug that directly into the inverter. And if you're in an area of the world where like the Philippines, where you get a lot of sunshine, depending on the season, I guess, you could run directly off of solar. It has 2 standard 120-volt power outlets to power other ancillary devices you might need, like a centrifuge or a mini centrifuge, if you're going to be doing that filtration. And then also the bioinformatics. So you need pretty decent spec computer to do the base calling and get the data into a format that is can be actionable and communicated to colleagues. Generating the raw data is one thing, but getting it into processing it and getting into an interpretable report in the field requires a lot of computer power. And so we've built in a mini computer that has 64 gigs of RAM, 2 terabyte solid state drive, GPUs with 2300 CUDA cores for base calling, and also the built-in software to do the variant calling, assembly. We can outfit it however you want, but Pangea is our metagenomics taxonomy classification software that we're actively sort of developing. We developed this a few years ago for a U.S. DTRA, so Defense Threat Reduction Agency funded program called Sample to Sequence, where we were building a sort of metagenomic biosurveillance workflow, sort of end-to-end optimizing the whole thing for, it was originally for the MySeq. We've been working doing a lot of work to try and port that over to Nanopore. And our group in particular, part of that role on that project was to develop the back-end bioinformatics. And so we wanted to not be command line driven, so you didn't have to have, expertise in bioinformatics and experience with working on the command line. You could just it's built into a point and click graphical user interface. And so this is sort of what it looks like. It's, as you analyze a sample, these sort of the taxonomy gets represented here. And in real time, you can sort of toggle these adjustments on the right, to identify, exactly what you might be looking for. If you're looking for SARS-CoV-2, you can type that into the organism name contains box, hit enter, and if that's in your sample, everything else will disappear. You can, filter things out a lot of times, especially if you're doing metagenomics. Oftentimes, you get a lot of stuff that you don't necessarily care about. And so sort of weeding through all of that is often hard to do quickly. And so, you can toggle these, adjust these things on the side here. So a minimum number of normalized reads, minimum depth of coverage, and all the organisms that don't meet those parameters will just sort of drop away in real time. So it's a nice way to sort of easily quickly parse through your metagenomic data. And we have that running on Mercury Lab. And then just think as simple as just a place to work. You know, you don't want to have to, you know, our early prototypes was really just sort of a camping table, and you know, we were going in and out of the backpack, and that's just not. It's not. You know, I kept knocking my tubes over and my pipettes over as I kept going in and out of the backpack, and you just the value of a laboratory workbench just becomes immediately. It's something that you really miss when you're out in the field and you're trying to do this. You know, the workflows are already hard and stressful enough. You really want to get good data. The last thing you want to be thinking about is, you know, the fact that your tubes are getting blown over in the wind, or you're kicking the side of your camping table. You know, just a good place to work really helps get this done efficiently and safely and reproducibly. And then also cold chain. So we built in we have a Credo cube. So these come in 4 degree or minus 20 degree versions. They're field stable for up to 96 hours. So they're essentially these cubes that have a phase change material in them. You stick them in the freezer. So for the minus 20, for example, you take those cubes, you stick them in a minus 80 freezer. And when you take those out and assemble the cube and put it in the insulated box, the inside of that cube will be stable at minus 20 for 96 hours. The 4 degree, you put in minus 20 degree freezer, you take it out, assemble it, and put it in the insulated box, and you have 4 degrees inside that cube for 96 hours. So that's, you know, really great for reagents and also samples if you want to take them back from the field. And yeah, just that ergonomic and stowable workbench that, you know, gives you a little bit of protection from the elements with the windshield, an LED light bar for working in low light, and spill containment features. So this is what it looks like. We're looking for folks to you know, partner with. So if anybody has interest in sort of you know, trying to deploy this type of platform, you know, in Mindanao or in any other, you know, locations in the Philippines, to try and do wastewater surveillance or any other type of infectious disease surveillance, please do reach out. And that's it. So happy to take any questions, and thank you for listening.
Thank you so much for that, Joe. We have a question in the chat box. Perhaps, if possible, Lara, if you could introduce yourself and ask your question. Maybe she's anyhow. If others have questions, maybe you could just mute yourselves.
Yeah, I do. Yes, Joseph, right?
Yes.
Yeah. So our group is actually doing a pilot investigation on wastewater surveillance here
Okay.
here in Davao City. The only challenge here compared to the US is that we don't have wastewater treatment plants, so we have to directly sample from, you know, uh, from the drainage in communities. So that's
Sure.
one thing that we're testing. That's part of our pilot testing. But I was really interested in your field. What do you call that? Field.
Mercury Lab.
The kit.
Yeah.
The field labs. Yeah. So,
Yeah,
such like
Lab.
something like a. Yeah. So, because I was thinking, eventually would like to, you know, if this works, if our pilot study works, eventually would like to expand the surveillance to other parts of Mindanao, just like what you mentioned. And we do have guests here from other COVID laboratories in other regions of Mindanao. And our like vision is to be able to connect with them and also introduce wastewater surveillance as a routine, like a complementary routine surveillance for COVID-19 in other
Sure.
areas. And we also have partners in Luzon. Actually, this is in collaboration with partners from the northern part of the Philippines in Luzon. They're also seeking for funds to have their work also funded in Luzon. And we're also working in collaboration with a group at North Carolina State University. So yeah, uh, the the modular lab that you're building, is this like straight from the sample? It does the extraction, the RT-PCR, and the sequencing all throughout?
Uh, so it's yes, it's whatever you want to plug into it from a device perspective. So Mercury itself, it doesn't come with a sequencer or the biomeme. You have to get those separately. But what it does have is sort of all of the ancillary logistics to support the use of those built in. So really what you see here is, we have a biomeme on the bench here. We have really close relationship, working relationship with the folks over there. And so, biomeme is, but it is sold separately, but, it comes with pipettes. This is this storage shelf is actually a removable backpack. So if you were, doing just PCR on a biomeme, but you wanted to go further afield than you want to drag the Mercury lab, you could pull all of this out and go walking farther afield. But, the lab itself has, the cold chain that you need, the Intel NUC, the GPU to do all of the bioinformatics, the power solution, the place to work, the workbench, all sort of built into an ergonomic environment. It's sort of approximating your laboratory workbench, but in the field. And then it's up to the user to pair with it, whichever device you want to use. So if you're doing, SARS-CoV-2 surveillance, with just PCR, you could just put a biomeme. There's several other options out there. We work with them closely. Or if you're doing sequencing, certainly, the MinION would be the genomic option. But
I see.
Yeah.
Okay. And I imagine you can expand this to other stuff that you want to detect. For example, aside from SARS-CoV-2, we are also looking at antibiotic resistance genes. So we can also do that, right?
Yeah, it's up. Yeah, whatever assay that you are, if you have it working on a portable PCR device, or if it's a genomics assay and it works on the MinION, yeah, absolutely, you could run it. We've used Mercury so far to do, our field tests have ranged from arbovirus surveillance in the Everglades. So we did a project where we were looking at Everglades virus, which is a strain of Venezuelan equine encephalitis virus. There's a species of mosquito in the Everglades that's the sole vector for the Everglades virus. And we teamed up with entomologists that were down there that were sampling that exact species. And we went looking for it with an early prototype of Mercury. And then also we did some work in the eastern shore of Virginia around chicken farms, looking for salmonella in the stream creeks around chicken farms. So yes, it's really it's up to the user to port whatever assay is relevant to you on those devices. But Mercury itself is just meant to keep you from doing it on sort of out of a backpack in an ad hoc manner that ends up, from experience, just ends up being stressful, and you end up not wanting to do that routinely. So this is meant to be some platform that can enable you to do that stuff in the field more routinely and reproducibly and safely.
Yeah. Another question. Biomeme, sure. Can it do multiplex RT-PCR?
Yes. So the device I showed in that picture, that was their original device, was the 2-3. So it was 3 wells, and you could multiplex 2 fluorophores per well. So you could run up to like 6 targets. The one shown here on the bench, that's their newest one, is the 3-9. So it's 9 wells, and you can multiplex 3 colors per well, so up to 27 targets. So yes, there are other devices out there. There's the MIC device that actually has, I think, 48 wells, and it's also multiplexable, per well. So it's a little bit bulkier.
Yeah, but when you run, for example, if you do MinION sequencing, this is going to take time, right?
Yes.
In the field.
Yeah. So depending on what you're doing, if you're doing, the Arctic protocol, it's a timely, it takes some time, but this isn't, you don't necessarily have to be in the field, but if you're in a location that just doesn't have, you
Yeah.
have shelter, but not necessarily like a laboratory environment, you could do it, in that kind of situation.
All right. Thanks.
Absolutely.
Other questions? I'll start reading the comments or going to the comments, and if there are no other questions that
Hello, good morning. Sorry.
Sorry.
My question is, in terms of the time frame from sample processing to sequencing, like, for instance, our concern is that sometimes because of the delay of the shipment of reagents, we acquire the sample much earlier, and it might take us like 1 month or 2 months in some instances before we can sequence the sample. So in terms of chances, like what's the likelihood of us getting the entire genome of the virus, like let's say 99% of the virus, or like, even if some segments are much informative, but if we are interested in, assembling the whole genomes from wastewater samples, like, is there a particular time period that we have to be able to sequence the samples for us to maintain those chances?
That's a good question. So I don't know. The one that one preprint was doing Illumina sequencing from a nanopore perspective. How long do you have to sequence to get sort of reliable coverage across the entire genome? I'm not actually sure. I think I believe it was in the neighborhood of 12 to 24 hours, but I would have to double check that. That's using the Arctic protocol where you're doing the amplicon. If you're doing shotgun, that's really sort of depends on how, what your titer level in your sample is. And, you could be sequencing for 48 hours plus before you got reliable signal, and probably wouldn't close the genome. I think to close the genome, you'd probably want to do the amplicon, the tiling approach. But I don't have a good answer for how, what's the necessary amount of time to get good coverage. I think it also sort of depends on the titer of the virus in your sample. And then also, especially if you're talking about wastewater surveillance, I mean, you have to keep in mind that, you could be, if you go in with the Arctic primers, those primers could be amplifying, different strains of the virus. And so you got to sort of be careful that you're not, assembling a Frankenstein that doesn't actually exist in nature. Assembling a genome and making sure that it's was from a single virus would probably require a good bit of sequencing. But from a surveillance standpoint, just a variant surveillance standpoint, you could do that from the single amplicons.
Cool, thanks.
I didn't realize, I wasn't looking at the chat. I just saw some of the questions here. So how heavy is the Mercury Portable Lab? So fully outfitted, it's about 90 pounds. So what is that? That's in kilograms. I think it's like 35-ish, 40 kilograms.
Around 40.
Yeah.
45.
So you get charged overweight baggage fees, but it does check on an airplane. But it does get a little tiring if you're lugging it around an airport for an extended amount of time, or down a city block for a while. But it wheels really comfortably. But it is about 90 pounds fully loaded, and that's with the Credo Cube. Without the Credo Cube, if you don't need cold chain because you're going to be processing on site, that saves you about 15 pounds. So several kilograms.
And Dr. Urban also has a question.
Okay.
Yeah, sorry. I guess, do you hear me?
Yes.
Yes, perfect. Yeah, sorry, sometimes it doesn't work here. Sorry. Yeah, thanks a lot for the talk, Joseph. So
yeah.
I've myself worked with nanopore sequencing just for freshwater monitoring, but I've also gotten involved in the wastewater monitoring here in New Zealand right now. And I was really interested in what you were saying about the actual filtration process at the beginning, where you said that it has been shown that absorption extraction works best right now. And it wasn't quite clear to me if it works better now than actual PEG, if you actually, after PEG, extract the virus both from the solid and the liquid parts, or if it works more or less same.
Yeah.
Or it would be great if you could comment on that. Thanks.
Yes. So I read this, and I can send you the review that I pulled this from, but it was, if I remember right, it was if you paired it with the spin-con filter and then process the liquid and solid fraction together. Where was that? Yes. Oh, I'm sorry, no. The PEG concentration, if when processing the liquid and solid fraction together, showed higher effectiveness, but yes, the absorption extraction method with the Amicon filters was the highest performer in this particular review that I can forward you. Happy to email you. But they had tested a bunch of different methods, and that one for that, the MHV, which was a good sort of proxy for SARS-CoV-2 because it enveloped coronavirus, was the better method.
Okay, so it was even better than PEG applied to both outputs. Okay, that's really interesting. Yeah, it would be great to have that review. Thanks.
Sure.
And I actually have another question, if that's okay.
Yeah.
So I was wondering about like the shotgun sequencing approach that you showed. It seems to be research that has just been published, right? But I was wondering, I mean, obviously shotgun sequencing is also possible with the MinION, but if you have yourself benchmarked anything there, or like, because obviously with shotgun, you always need to sequence a lot more than what you're actually interested in. So what maybe the sequencing costs are and so on, as compared to the qPCR approaches that you're using at the moment.
Yeah, so I mean, I think I would think that shotgun would probably be more, it's more direct and it's less biased, but it would probably be less effective, I think, routinely. It's a little bit of luck there, and I have experience with a little bit of that luck. Like I mentioned on our Everglades study, we were looking for Everglades virus. We wanted to see if we could do it from a shotgun standpoint. We went looking for it with a PCR assay, and we actually detected it. Usually the folks that we were with said, you know, maybe 1 out of 100 mosquito pools you'll actually detect virus. We only processed 50, and we got one of them that came up hot for the virus from a PCR assay, and on the biomeme. And then we went back to that sample and did you know, a cDNA conversion and then just metatranscriptomic sequencing just to see if we could, in that context, also reproduce that signal. And we were able to on the MinION. We did it on the MiSeq, Illumina MiSeq, and the MinION. And you know, of course, on the MiSeq we got much better coverage. You know, this was also in 2016, so back when, you know, MinION was, the flow cells weren't as good as they are now. But even back then, we were able. So we found. I think it was 16 high quality variants in our virus, and 10 of those, if I remember right, we were able to detect on the MinION from a shotgun sequencing. So that was, you know, pretty encouraging that we were able to get that level of signal nucleotide variant detection from shotgun sequencing. So flow cells have gotten better and since then, and so it's definitely doable. But yes, we sequenced for I think it was 16 hours to get that back then. So you know, if you can amplify, if you know what you're looking for and you have an assay and you can amplify it and then sequence it, I think that'll always just be better. But you know, obviously shotgun sequencing, if you don't know what you're looking for, you don't have an assay for it, you know, it is doable.
Yeah, thanks a lot. Yeah, that makes a lot of sense. Sorry if I missed that, but is there already an assay that includes the latest variants, like the UK and the South African variant? Do you know about anything like that?
As far as I understand, the latest Arctic protocol does not have any dropouts from those variants, because I think where the primers sit are outside of where the variants are. So I don't think that they've been impacted. Don't quote me on that, but from my reading and keeping the thumb on the pulse of Twitter, I haven't seen that's been an issue yet. But certainly it's definitely something that needs to be continuously monitored.
Thanks a lot.
By the way, Dr. Urban will be our guest next week in our talk next Friday next week. And her paper just came out at eLife, so I'll be sending it out later today.
Cool.
Thanks a lot, Khalil. Yeah, some
Looking forward to
promotion.
it. Any other questions?
Hello, I have a follow-up question regarding the storage. The one you mentioned regarding the effect of crystalline formations at -20, is it specific for viral particles, or does this also affect viral nucleic acids? Because here, not all institutions has capacity for -80. So sometimes they use the next one, -20, as a proxy.
Mm-hmm.
Like in terms of comparison, if we don't have -80, like is 4 degrees better, or should
I think
we opt for -20?
if you're able to process, relatively quickly, I think it would probably be better to keep it at 4 degrees. If
4
you don't want
degrees.
to, yeah, if you want to do long-term storage and you don't have a -80, yeah, -20 could be problematic for if you're trying to do molecular stuff. And then from what my reading of that paper, it impacted the actual genomic material, not just the viral particle.
Okay, thank you. This is new for us. Thanks.
Yeah, I wasn't aware of that either, actually. But and I can send you the paper from that too. That I found that. Was,
yeah, I would love to read the paper. Thank
sure.
you.
Yeah, Khalil, I'll forward them to you, and then I'll let you. Or actually, I think I have a lot of people's emails from the Zoom invite, so I can reply all to that with those papers.
Yeah, just reply to the invitation.
Yeah.
Any other questions? All right, so I guess, well, when you reply to those invites, we also have your email, so we could continue a conversation over email.
Yes.
Yeah. Could you also reach Joe over Twitter? So if you're present there. All right.
Yes.
So
Khalil,
yes.
do we have a recording of this?
Yes, I'll be posting it. So and then all the trainings are available at the website. Just go to accessiblegenomics.org, and then you'll find a link there to all the trainings. Yeah, we plan to create a resource which could be used by other locations that want to set up a genomics layer to their existing lab. So that's the output of this project. So yeah. All right. So thank you so much, Joe, and thank you so much to all the participants for joining us. And
Yes.
yeah, so I'll see you guys around.
Alrighty.
Bye.
Take care.
Thank you. Thank you for the talk. It's
you.
wonderful.
Thank you. Bye.
Bye.